Hi, everybody. Today's focus is going to be on blood typing. So we're going to learn how you identify somebody's blood type by looking at the antigens that are present on the surface of the blood, and also how do you identify someone's blood type when basically you have no information about that person's blood type or antigens that are found on the surface of the red blood cells. So let's go ahead and get started. So when we're looking at the blood typing concept, two words needs to be your friends. One is the word antigen and the other one is antibodies. So let's just start by looking at antigens. So antigens are these proteins that are found on the surface of your red blood cells. And they're extremely important because they're the ones that are basically going to help us identify our blood type. So you probably have heard of blood types like A positive, B negative, O negative, AB positive, A negative, basically various versions of the blood type. So those letters that we see, the A's and B's or the combination of A or B, and the blood being positive or negative comes from the presence of these antigens on the surface of your RBC. So there are three main antigens that are found on our blood type or on our red blood cells that are help us identify our blood type. And these are antigen A, antigen B, and antigen Rh. Now, if you have A, your blood type is basically A. If you have B, your blood type is B. If you have both A and B, your blood type would be AB. The Rh components or antigen is the one that basically makes your blood positive or negative. So if Rh is one of the antigens on your RBC surface, then your blood type is positive. However, if Rh is absent from your RBC surface, then your blood type is negative. So it's the presence or absence of Rh that makes your blood positive or negative. Now, what if I have no A or B on the surface of my blood type? On the surface of my red blood cells. For those, we refer to those as blood type O. And again, if the Rh is present, then your blood type would be O positive. And if your Rh is absent, your blood type would be negative. So an example of identifying your blood type. If you only have antigen A on the RBC surface, then your blood type would be A negative. Next, let's go ahead and take a look at our antibodies, which is the second component you need to be familiar with. Now, antibodies are found on the surface of, antibodies are found inside your plasma, and their goal is to identify any foreign antigens that are associated with your red blood cells. And upon identifying those antigens, they will trigger what we identify as an immune response. So antibodies are basically part of what we identify as your immune response or your lymphatic system. And their job is to basically identify components that are foreign or unknown to us and destroy them. Now, like antigens, we also have three different types of antibodies. And their options are going to be still what? Anti-A antibody, anti-B antibody, and anti-Rh antibody. So notice that the options are still the same options we have for our antigens. Again, A, B, and Rh. I'm going to make a quick note for you here right now about the Rh. So normally, nobody has Rh antibodies or anti-Rh antibodies in their blood. However, if your blood type is negative and then you get exposed to somebody that has a positive blood type, which means they have Rh in their blood, then at that case, your body automatically starts to produce anti-Rh antibody. But under normal condition, regardless if you are negative or positive blood type, you will not have anti-Rh antibody in your bloodstream. So that doesn't hold true for anti-A and anti-B, as I'll talk about it in just a minute. Now again, what is the function of our antibodies? Antibodies' goal is to identify foreign antigens that are not supposed to be there, and that causes an immune response, which appears as an agglutination, which basically means it's going to form clumps of your red blood cells. It causes your RBC to basically form these clumps or these larger structure by holding them together and kind of gluing them together. Kind of remember, this is a really bad idea if the idea of your blood is to be fluid and now you have these kind of clumps or agglutinates within your bloodstream. So that can cause severe damage to the organs and to the blood vessels, and in many cases can lead to death if it's not treated. So what is the basically matching of the antibody with antigen? If you have anti-A antibody in your plasma, then that anti-A antibody will recognize the corresponding antigen, which is AGA. AG is shorthand for antigen. So, do I want to have this combination under normal condition for my blood? The answer is no. So, if a person has an antigen A on their bloodstream, they should not have anti-A antibody in their plasma. Think about this, guys. If you have anti-A in your plasma, an antigen A on your red blood cell surface, that means that antibody can attack your own red blood cells and cause them to clump or agglutinate and basically cause major health issues for you. So if I have antigen A on my red blood cell surfaces, I do not want to have anti-A antibody in my bloodstream. However, I do want anti-B antibodies in my bloodstream, because then that is known as an unknown or foreign antigen. So if I get introduced with a blood that is B blood type, then I want to be able to identify that as foreign. So again, you do not want your own body to attack your own cells or your own red blood cells. So you should not have a match between your anti-A, you shouldn't have a match between your antigen and your antibodies at a given time. You only want to have this scenario where you have an unknown blood type getting introduced to your bloodstream. Let's look at an example. So hopefully this will make it a little bit clearer. So let's say Misty has antigen A on their red blood cell surface. First question is, what is Misty's blood type? So take a moment. Antigen tells me my blood type. So if I only have A on the surface of my blood cell sample, on the surface of my red blood cells, then my blood type would be A negative. A negative, the negative component comes from the fact that I have no Rh antigens. So what is the antibodies, or what are the antibodies that are found in the plasma? So if it should not be A, because A is already in the antigen, so your option would be B and Rh. Another way I tell my students to think about this is, do this for yourself, and hopefully again this will make it a lot easier for you to understand. The options for antigen and antibodies are the same, right? Your three options are B, A, B, and Rh. So I'll tell students, let's say you're working on your antigen, and I already know my antigen is A. So I'm gonna scratch this from my options because it's already been used. So what is left out would be my options for antibodies. So here, B and Rh. Let's look at another example. What if I say the person's blood type, pay attention to this, guys. The person's blood type is B positive. And I ask you, what is the person's antigen? Again, antigen is giving me my blood type. So my antigen options would be B. And because it's positive, then I will have RH as my antigen, correct? Now again, rewrite the three options you have for antigen and antibody. I have used B and RH as my antigen, so I can draw lines on those and basically say those are out. What am I left with is what would be identified as my antibody. So antibody would be A. Just giving you another example, I want to make sure you guys are comfortable with this. Let's say I told you that your blood type is O positive. And I ask you, tell me your antigen. Again, O means you have neither A nor B. However, because you have this plus sign right here, it means you do have RH as your antigen. So again, write the three options for yourself. The one I use for antigen is only RH, so I'm gonna draw an X on that one. Whatever is left out would be my antibody, which would basically be A and B. A quick note, guys, when you're identifying your blood type, you always have either A, B, and then a positive and negative, right? Or O. When you're talking about antigen or antibody, you cannot have a positive or negative, right? You will have the option of RH either being present or absent. And notice again, the letters are going to be separated by a comma, so you cannot write your antigen is AB. You cannot write your antibody is AB. You have to write your antigen is A, comma, B, or your antigen is A, comma, B. Do not put the letters together because that's how we identify blood type, not how we write antigens or antibody. Now here is an overview of all the blood types that we have in terms of blood types A, B's, and O's. This is kind of helping you identify the antigens, the blood type using those antigens, and the possible antibodies. And I'm gonna kind of go over this with you to make sure everybody's on the same page. If I have only antigen A, your blood type is A negative. Again, no RH, so it is negative. And among the options that I have, B and RH will form my antibodies. If I have A and RH, my blood type becomes positive. And the only one that I didn't use among the three options would be B for my antibody. If I have antigen B, my blood type is B negative. And the two options remaining, which is A and RH, would be my antibodies. B and RH together form my B positive blood type with an antibody being A. A and B give me a B negative with an RH being my antibody. And if I have all three present, A, B, and RH, that's gonna give me A, B positive. And because I use everything as antigens, then there is no antibody. So no antibody. If I only have RH, my blood type is O positive, and A and B would be my antibodies. And if I have no antigen, so basically your red blood cell is bare, then your blood type is O negative, and all the three options will be identified as your antibody. Now, AB positive and O negative are important because they're identified as universal donors and universal receivers. So universal receiver is your blood type AB positive. So AB positive has basically all the options as antigens, therefore it has no what? Antibody. So there is no line of defense that's going to come in and say, oh, you are foreign material, I'm gonna kill you by clumping it, right? So universal receiver basically can accept every possible blood type regardless of if it's O negative, O positive, A negative, etc. It receives all of those blood type because basically there is no guards that come and tell you, you can't come in. If you come in, I'm going to kill you. Opposite of universal receiver is universal donor. Universal donor is O negative. So going back to your O negative, notice O negative has no antigens. So there is nothing on its surface. So it can give to everybody, right? So it's happy to basically, it's very selfless. It can give to everybody because it says, I have nothing that you guys can identify as foreign and trigger an immune response against it. So O negative will be identified as my universal donor as it can give to all blood type. However, there is a problem with a person being O negative. They can only and only accept from an O negative blood type. So if an O negative individual requires a blood transfusion, the only way it can get that blood transfusion is from what? An O negative individual. Because otherwise, the antibodies they have, either A, B, or Rh, will attack the blood type and cause major damage. Here's another way of depicting the same information. This one doesn't have the Rh depicted in it, but it still gives you a good idea about the main groups of A, B, and O groups. So here's a little more of visualization. The structures you see on the surface of your red blood cells are antigen A and B. These are the antibodies that are found in the plasma. And again, notice they are opposite of each other, right? So if your blood type is A, your antibodies are B, and vice versa. And then here's the list of your antigens, which is basically visualized on your RBC. Now, the next concept we're going to focus on is, what if a patient walks into the hospital and they need a blood transfusion, but the nurses have no idea what that individual's blood type is? So the reason I can tell you the blood type so far is because I am telling you what type of antigens are on the RBC surface. And based on that information, I can figure out my blood type. But how would I know what antigens are on the surface of somebody's blood so I can identify the blood type? So for that, we use something called a blood typing kit. Now, there are different versions. Of course, the hospital uses the, I guess, more hands-on, pretty fast techniques to use to figure out somebody's blood type. But regardless of what type of blood typing kit you use, the procedure would be very similar. The components would be very similar to what you would be carrying and would be observing in the experiment here. So what does your blood typing kit contain? It typically contains these small plastic trays that has 3 little wells on them, and each of the wells correspond to the antigens, either A, B, or Rh. We have synthetic anti-A, synthetic anti-B, and synthetic anti-Rh antibodies. Again, this is not something you will find inside your bloodstream. This is something that we are adding on top of your blood. And then you have these wooden sticks that is going to help you mix your blood sample with the antibody serum. Here's basically what I'm talking about. Here is your little tray with the 3 wells in them, letter A, Rh, and B. And here is a solution for anti-A serum or anti-B or anti-Rh anti-serum or antibody. And this is basically what you use to mix your sample. Now, what are you exactly looking for when you do these blood typing kits? When you start your sample of your blood, the blood is pretty clear. So again, this might be varied. It depends on the kit. But what you usually start is a pretty liquid or fluid sample of blood, which kind of makes sense. Now, if there is some sort of reaction where an antibody, this synthetic antibody, recognizes an antigen or its corresponding antigen, then that forms an immune response. So you will be visualizing agglutination or clumping of the antibody with the corresponding antigen. And that interaction basically helps you figure out what your antigen is. And based on that antigen, you can figure out what your blood type is. So let's look at some of the examples before I kind of direct you into the video and walk you guys through the actual experiment. So let's say you get an unknown blood sample and you want to figure out what this blood type is, the person's blood type is. Here is my little tray. Sorry for the messy drawing. Again, the location of these are not identical, but you get the point. So I'm gonna add an unknown blood sample to each of these. This is the same blood sample. So this is, let's say, Mr. X blood sample, doesn't matter. So we adding the same blood sample from Mr. X to each of these three wells. Now the one that has A written on the top, what I would be adding to it is anti-A antibody. To the one that is B, I'm gonna add can't spell apparently, anti-B, and then RH, I'm gonna add anti-RH antibody. What I'm gonna do next is literally take these blood samples and mix them with their antibody. And I am looking for any changes that may be identified as clumping or agglutination. So let's say I have done this, and the clumping that I've seen is visual in this sample and visual in this sample. So there are visible changes, in both A and RH, and there is no change in B. So what does that exactly mean? So imagine this is my red blood cell, okay? This is Mr. X's red blood cells, okay? So what it says is anti-A serum or anti-A antibody recognized an antigen on the surface of the blood. Now, anti-A can only identify antigen A, which means Mr. X will have to have antigen A on their blood type. So here is a bunch of A's on its surface. And again, these are representative of antigens. Antibody B stayed the same, didn't recognize anything, which means there is no antigen B for antibody B to be recognizing. So there is no B on the surface of the RBC. RH, however, the anti-RH antibody recognized what? Antigen RH, and therefore triggered an immune response or clumping. So what that tells me is there are antigens RH on the surface of my red blood cell for this sample. So I have, based on this information, A and RH as my antigens. And now that I have my antigens, I can say what is my blood type. So based on what you have, your blood type is A positive. What if I say, what is the antibody you will find in the plasma, not the antibody that is on this screen? These are synthetic antibodies. What are the antibodies that will be found inside Mr. X's plasma? And remember, if I use two options of the antigen, the only option for antibody would be B. Let's take a look at another example, and you guys can try to follow this on your own and see if you can provide an answer for it. So here is another tray, same concept as we just talked about. So let's say this is Mrs. B. So again, three wells, A, B, and C. Oh, sorry, that's A, B, and C. Sorry. A, B, and RH. I have added Mrs. B's blood type or blood sample to A, B, and RH. And I also added, so I'm gonna say blood, blood, blood. And I'm also adding I want to make sure you guys understand this. This is a synthetic anti-A that I'm adding. It is not what you found in your body. This is a synthetic versions in the, basically a tube that you are adding to the sample. And then anti-RH antibody. And I'm looking at this sample, and the only one, let's say, that is clumping would be my B. So my B is clumping after I have mixed the sample. But there is no change, so I'm just gonna kind of highlight this. But there is no change in my A or RH well. So again, I'm gonna draw my little red blood cell. So this is Mrs. B, RBC. Now, the anti-serum that I had for the blood sample did not mix or identify any antigen. Therefore, anti-A did not identify antigen A. No antigen. Same thing can be true for the anti-RH antibody. Didn't recognize anything, no clumping. But you did see B clumping, which means antibody B recognize antigen B. So I'm gonna draw my antigen B right here. So if I have antigen B, what is my blood type? My blood type would be B negative. And if I ask you what are my antibodies then, you only use B as your antigen, so A and RH would be your antibody. So that is it for our understanding in terms of how we would use a blood typing kit to identify unknown blood types. Do remember that this is an interaction between an synthetic antibody and not what is actually happening in your body. So a lot of people cannot make that distinction because as soon as they hear the word antibody, they automatically think about plasma and how they should not be matching with their antigen. But please do keep in mind that this antibody is not the same as the antibody found in your blood sample. So you have a video to watch, and this video has examples of 4 blood samples. These are synthetic blood samples as well. So I have done and run the experiment for you. So what you will be looking at is trays of synthetic blood that is mixed with antibodies. And based on the changes you observe in the tray, you would basically be carrying the same process that I've done here and identify the person's blood type based on the clumping that are visible in each of the wells for each of the samples. And make sure you guys submit the assignments. And if you guys do have any questions, don't forget to email me.